The next section is the biochemistry of the cell. This part of the lecture deals with the different components of the cell, specifically the organelles and their respective properties, and more especially, their functions. We will also be including in our discussion certain biochemical aspects of these components, and the different biochemical pathways contained therein. The cell, coming from Latin word cella, meaning small room, is the basic structural, functional, and biological unit of all known organisms. A cell is the smallest unit of life, the smallest entity that is capable of metabolic processes. Additionally, cells are often called the building blocks of life. Additionally, cells, though microscopic, are regarded as the complex unit of life, since they are highly organized entities consisting of separate and distinguishable parts, each performing important functions in the overall living process. There are two types of cells: eukaryotic, which contain a nucleus, and prokaryotic, which do not. Let us now go to the different components or organelles that comprise the cell. The first of this is the nucleus. The nucleus is a membrane-enclosed organelle found in most eukaryotic cells. It contains most of the cell's genetic material, organized as multiple long linear DNA molecules, in complex with a large variety of proteins such as histones to form chromosomes. The genes within these chromosomes make up the cell's nuclear genome. The function of the nucleus is to maintain the integrity of these genes, and to control the activities of the cell by regulating gene expression. Let's now look more closely at the structure of the nucleus. The main structural elements of the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and keeps its contents separated from the cellular cytoplasm. And the nuclear lamina, a meshwork within the nucleus that adds mechanical support much like the cytoskeleton supports the cell as a whole. It is a double membrane containing phospholipids, which serves to segregate the nucleus from the cytoplasm. The inner nuclear membrane defines the nucleus itself, and the outer nuclear membrane is continuous with the rough endoplasmic reticulum. The space between the inner and outer nuclear membrane is continuous with the lumen or inner cavity of the rough endoplasmic reticulum. The nuclear pores. Because the nuclear membrane is impermeable to most molecules, nuclear pores are required to allow movement of molecules across the envelope. These pores cross both membranes of the envelope, providing a channel that allows free movement of small molecules and ions. The movement of larger molecules, such as proteins, is carefully controlled, and requires active transport facilitated by carrier proteins. Nuclear transport is of paramount importance to cell function, as movement through the pores is required for both gene expression and chromosomal maintenance. The nucleolus is a very electron-dense suborganelle of the nucleus, easily recognized under the light microscope. The current belief is that the nucleolus is the site within the nucleus where ribosomal RNA biosynthesis occurs. The nuclear organizer, a region of one or more chromosomes in the nucleolus, contains many copies of the DNA that direct the synthesis of ribosomal RNA. The nucleolus is roughly spherical, and is surrounded by a layer of condensed chromatin. No membrane separates the nucleolus from the nucleoplasm. The nucleoplasm. Throughout the nucleoplasm, considered to be the non-nuclear region of the nucleus, there appear regions of less electron density, particularly near the nuclear membrane. These regions are termed chromatin, and are known to contain the major portion, 95% or more, of the total DNA found in the cell. Functions of the nucleus. The nucleus is the site where genetic information is stored as DNA. It is also the site of DNA replication, thus ensuring the perpetuation of the cell line. This is also the site where genetic information is transmitted from to the rest of the cell. The next organelle in our discussion are the mitochondria. These are rod-shaped particles with a length of 1.5 to 2.0 micrometers. The mitochondrion is approximately a 20th of the size of the cell nucleus. Although mitochondria are found in virtually all eukaryotic cells, the size, the shape, and number vary from cell to another in response to shifts in metabolism and as a result of cell aging. Certain generalizations can be made regarding the number of mitochondria per cell. In cells characterized by a high degree of aerobic, or oxygen-dependent metabolism, the number per cell might be quite large. In contrast, cells participating primarily in anaerobic, or non-oxygen-dependent metabolism, contain only a few mitochondria. Like the nucleus, the mitochondria is bounded by a double membrane, two unit membranes, separated by an intermembranous space. The two membranes, however, have different properties. Because of this double-membraned organization, there are 5 distinct compartments within mitochondria. There is the outer membrane, the intermembranous space, or the space between the outer and inner membranes, the inner membrane, the Christie space, which is formed by enfoldings of the inner membrane, and the matrix or space within the inner membrane. The outer mitochondrial membrane, which encloses the entire organelle, is smooth and continuous. It has a protein to phospholipid ratio similar to the eukaryotic plasma membrane, which is about 1:1 by weight. The outer mitochondrial membrane contains numerous integral proteins called porins, which contain a relatively large internal channel measuring about 2 to 3 nanometers, that is permeable to all molecules of 5,000 Daltons or less. Larger molecules can only traverse the outer membrane by active transport through mitochondrial membrane transport proteins. The outer membrane also contains enzymes involved in such diverse activities, as the elongation of fatty acids, oxidation of epinephrine, and the degradation of tryptophan. Let's now proceed to the inner mitochondrial membrane. This membrane undergoes an extensive and irregular folding within the mitochondria. The inward folds are called Christie. The fluid-filled interior of the mitochondria is called matrix or central space. The inner mitochondrial membrane contains more than 100 different polypeptides, and has a very high protein to phospholipid ratio, of more than 3:1 by weight. Additionally, the inner membrane is rich in an unusual phospholipid known as cardiolipin. Unlike the outer membrane, the inner membrane does not contain porins, and is highly impermeable. Therefore, almost all ions and molecules cannot pass through the inner membrane. They will be requiring special membrane transporters to enter or exit the matrix. In addition, there is a membrane potential across the inner membrane. The inner mitochondrial membrane is compartmentalized into numerous Christie, which expand the surface area of the membrane. The inner mitochondrial membrane serves a diverse array of functions. First, it is the site of aerobic metabolism of the cell, which includes the crucial biochemical processes of the citric acid cycle and oxidative phosphorylation. It also houses the enzymes that catalyze the final oxidation of sugars and beta oxidation of lipids, and of course, the synthesis of ATP from ADP and inorganic phosphate. These different activities produce nearly all the energy required for the growth and viability of the cell. This is the primary reason why the mitochondria is regarded as the powerhouse of the cell. The intermembranous space is the space between the outer and inner membranes. It serves a special role in the electron transport chain. The matrix, on the other hand, is the space enclosed by the inner mitochondrial membrane. It contains a highly concentrated mixture of hundreds of enzymes, in addition to the special mitochondrial ribosomes, tRNA, and several copies of the mitochondrial DNA genome. The matrix contains 2 to 4% of the total DNA of the cell and ribosomes. The mitochondrial DNA contains genes coding for some of the proteins contained in the mitochondria. Because of these, the mitochondria can be considered a sort of secondary mini-cell. This is a mitochondrion, as seen with an electron microscope, which shows a cross-section of the organelle. This image, on the other hand, shows a three-dimensional representation of the arrangement of the mitochondrial membranes. Note the smooth outer membrane, and the highly convoluted inner membrane. This inner membrane contains most of the proteins responsible for cellular respiration, and it is highly folded to provide a large surface area for this activity. In the schematic cell shown here, the interior space of the mitochondrion is colored. This slide closes our discussion on the mitochondrion, and we are now ready to move to the next organelle. Let's now look at the endoplasmic reticulum. The endoplasmic reticulum is a net-like system, or reticulum, of flattened, membrane-bound regions localized within the cytoplasm. Unlike the nucleus and mitochondria, the endoplasmic reticulum is not a singular, highly ordered entity, but an irregular and interconnected array of membranous vesicles. It is part of the endomembrane system. The basic structure and composition of the ER membrane are similar to the plasma membrane. Found in all eukaryotic cells, the endoplasmic reticulum is responsible for several specialized functions such as protein translation, folding and transport of proteins to be used in the cell membrane, or to be secreted from the cell, the sequestration of calcium, and production and storage of glycogen, steroids, and other macromolecules. Two types are known: the rough endoplasmic reticulum, also known as RER, and the smooth endoplasmic reticulum, or SER. The rough endoplasmic reticulum. The surface of the rough endoplasmic reticulum, or RER, is studded with protein manufacturing ribosomes, giving it a rough appearance. Hence its name. But it should be noted that these ribosomes are not resident of the endoplasmic reticulum incessantly. The ribosomes only bind to the endoplasmic reticulum once it begins to synthesize proteins destined for sorting. The ribosomes attach to the RER functions in the biosynthesis of certain membrane and organelle proteins, and other proteins to be secreted from the cell. It should be noted, as well, that the membrane of the rough endoplasmic reticulum is continuous with the outer layer of the nuclear envelope. Although there is no continuous membrane between the rough ER and Golgi apparatus, membrane-bound vesicles shuttle proteins between these two compartments. The rough endoplasmic reticulum works in concert with the Golgi complex to target new proteins to their proper destinations. The smooth endoplasmic reticulum, or SER, consists of tubules and vesicles that branch, forming a network. It is characterized by the absence of attached ribosomes, hence the term smooth. The SER network allows increased surface area for the action or storage of key enzymes and the products of these enzymes. The smooth endoplasmic reticulum has diverse functions in several metabolic processes, which are stated as follows. The storage, in the cisternal space, and transport of proteins, that are destined for secretion from the cell. The site of synthesis and metabolism of fatty acids and phospholipids. The site of enzymes that modify or detoxify chemicals and carcinogens, by converting them into more water-soluble conjugated products that can be secreted from the body. When whole cells are disrupted utilizing various methods, the reticulum network is randomly cleaved, and smaller fragments are eventually isolated. These reticulum pieces are termed microsomes. Additionally, it is worthwhile to note that the smooth endoplasmic reticulum is known for its storage of calcium ions in muscle cells, and as such will be called the sarcoplasmic reticulum. Here is a last look at the endoplasmic reticulum, or ER. This is the schematic diagram of an animal cell showing the endoplasmic reticulum, as shown in green. On the other hand, this bigger image is an electron micrograph of a thin section of a mammalian cell, showing the endoplasmic reticulum. Please note that it is continuous with the membrane of the nuclear envelope. The black particles, studding the particular region of the ER shown here, are the ribosomes, the molecular assemblies that perform protein synthesis. The next organelle in our list is the Golgi apparatus. Also called the Golgi body, Golgi complex, or dictyosome, it is an organelle found in typical eukaryotic cells. It is typically comprised of a series of 5 to 8 cup-shaped, membrane-covered sacs, called cisternae, that look something like stacks of deflated balloons that are localized near the nucleus. The primary function of the Golgi apparatus is to process and package macromolecules synthesized by the cell, primarily proteins and lipids. It is often referred to as the distribution and shipping department of the cell. Each Golgi stack has two distinct ends, or faces. They are the cis and the trans faces. The cis face of a Golgi stack is the end of the organelle where substances enter from the endoplasmic reticulum for processing. On the other hand, the trans face is where the processed substances exit in the form of smaller detached vesicles. Consequently, the cis face is found near the endoplasmic reticulum, from whence most of the material it receives comes. In turn, the trans face is positioned near the plasma membrane of the cell, to where many of the substances it modifies are shipped to. Cells synthesize many different macromolecules required for life. The Golgi apparatus is integral in modifying, sorting, and packaging these substances for cell secretion or for use within the cell. The functions of the Golgi body are myriad. Let us enumerate them. First, it accepts proteins from the rough and smooth endoplasmic reticulum system for concentrating and packaging into dense granules which are then secreted from the cell. Next, it is also responsible for concentrating certain proteins and packaging in membrane-bound bodies, such as lysosomes, which remain in the cell. Additionally, it also aids in the post-translational glycosylation and the addition of lipids to proteins. To continue with the other functions of the Golgi body, it is also implicated as the cellular site for the biosynthesis of complex carbohydrate materials which are ultimately secreted and deposited in the exterior coating of the cell. Because of these various crucial processes, the Golgi body is also known as the traffic police of the cell. It plays a key role in sorting many of the cell's proteins and membrane constituents, and directing them to their proper destinations. A tangible example is the Golgi body's role in modifying secretory proteins, for instance, by glycosylation or phosphorylation, and packaging, and directing them to their proper sites in the cell. A last look at the Golgi apparatus. This is a schematic diagram of an animal cell, with the Golgi apparatus colored red. The next is a drawing of the Golgi apparatus reconstructed from electron microscope images. It is composed of flattened sacs of membrane stacked in layers, from which small vesicles of membrane pinch off. Only one stack is shown here, but several can be present in each cell. And this last image is the electron micrograph of the Golgi apparatus from a typical animal cell. Microbodies are small compartments found in the cytoplasm, which are surrounded by a single membrane. Nearly all eukaryotic cells contain variable numbers of microbodies ranging from 10 to 1,000 per cell. They contain a high concentration of certain proteins and enzymes, and appear as dense regions in electron microscopy. Microbodies are formed by pinching off as buds from either the Golgi body or from the rough endoplasmic reticulum smooth endoplasmic reticulum network. Microbodies include lysosomes and peroxisomes, which we will be discussing in the next few slides. Lysosomes are membrane-limited organelles that contain various types of enzymes, also known as acid hydrolases, that degrade polymers into their monomeric subunits by catalyzing the hydrolysis in these polymers. Examples of these acid hydrolases are phosphatases, nucleases, and proteases. These enzymes work only at acid pH, and are inactive at the neutral pH values of the cell, and most extracellular fluids. To enable these enzymes to function, the inside of a lysosome is maintained at about pH 4.8 by a hydrogen ion pump in the lysosomal membrane. The acid pH helps to denature proteins, and make them accessible to the action of the lysosomal hydrolases, whose structures resist acid denaturation. If a lysosome releases its contents into the cytosol, where the pH is between 7.0 and 7.3, no degradation of cytosolic components takes place. Here is an illustration of the various examples of acid phosphatases that are contained within the lysosomes. Take note that these enzymes act on almost all the biomolecules, including carbohydrates, proteins, lipids, nucleic acids, and other molecules containing phosphate ester bonds. To continue, two types of lysosomes are identified. The first is the primary lysosome. This type of lysosomes is roughly spherical in shape and are considered newly formed, and have not yet engaged in any cellular activity. These lysosomes are known to be in the latent phase, and do not contain obvious particulate or membrane debris. On the other hand, secondary lysosomes are larger and irregularly shaped and contain particles and membranes that are being digested. These type of lysosomes appears to result from the fusion of primary lysosomes with other membrane-bound organelles. Additionally, the content of these lysosomes seems to be released as a package into the phagoacetosome, when they fuse to form the phagal lysosome. Secondary lysosomes contain both hydrolyses and substrates. This is an illustration of the degradation of exogenous and endogenous macromolecules in the lysosome. As you can see, this illustration just reiterates the functions of lysosomes: first, the degradation of aged or defective organelles. And secondly, the degradation of extracellular macromolecules brought into the cell by endocytosis. Feel free to pause the presentation to trace the degradation of the different macromolecules and their corresponding products. Moving on, peroxisomes are a class of small, membrane-limited organelles found in the cytoplasm of all animal and many plant cells. These organelles contain various enzymes that degrade fatty acids and amino acids, reactions of which produce hydrogen peroxide. As mentioned, peroxisomes contain enzymes that degrade fatty acids and amino acids. A byproduct of these reactions is hydrogen peroxide, a corrosive substance that oxidizes many amino acid side chains, and may cause cellular damage if in excessive amounts and left unchecked. Fortunately, the peroxisomes also contain copious amounts of the enzyme catalase, which catalyzes the conversion of toxic hydrogen peroxide to water and oxygen. Catalase is one of the scavenging enzymes that protect the cells from oxidant damage. The cell membrane, also called the plasma membrane or plasmalemma, is a selectively permeable lipid bilayer which comprises the outer layer of a cell. It is continuous with the nuclear membrane and other intracellular membrane of various organelles. The cell membrane is a semi-permeable barrier that defines the outer perimeter of the cell. It allows nutrients to enter the cell, and filter out unwanted materials in the extracellular milieu. This is the function of transport proteins and permeases found in this membrane. The plasmalemma also prevents metabolites and ions from leaving the cell, thus playing a major role in maintaining the proper ionic composition and osmotic pressure of the cytoplasm. And lastly, the membrane serves in the communication and interaction among cells, though gap junctions and receptors found in the membranes. This is a simplified diagram of the cell coat, also known as the glycocalyx. As defined, the cell coat is a viscous layer that covers the cell membrane or cell wall. This structure consists of the oligosaccharide side chains of glycolipids and integral membrane glycoproteins. It is also made up of the polysaccharide chains on integral membrane proteoglycans. In addition, adsorbed glycoproteins and adsorbed proteoglycans contribute to the glycocalyx in many cells. Note that all of the carbohydrates is on the non-cytoplasmic surface of the membrane. The cell coat serves a wide array of functions. First, it is crucial for cell-to-cell recognition and for tissue reorganization. It also acts as filters to regulate the passage of molecules, thus, a highly effective diffusion barrier. And lastly, it plays a major role in cell-to-cell communication, like contact inhibition, for instance. The cytoskeleton is a cellular scaffolding or skeleton contained, like all other organelles, within the cytoplasm. The cytoplasm in eukaryotic cells contains an intricate assembly of microtubule structures, all connected to a thin thread-like structure called microtrabecular strand. This lattice may also connect with other tube-like structures called myofilaments, which are associated with the cell membrane. All consist of protein molecules. The entire assembly is termed a cytoskeleton network, a highly structured network of proteinaceous filaments. The cytoskeleton elements have important functions. Their dynamic structures maintain overall cell shape, and also has been known to protect the cell. These elements also enable some cell motion and play important roles in both intracellular transport, for instance, the movement of vesicles and organelles. It is also important in facilitating cellular division and providing surfaces on which some chemical reactions may occur. Lastly, the cytoplasm, also known as the cytosol, is the internal fluid of the cell. A significant list of cell metabolic processes occurs here. Important pathways, like glycolysis, gluconeogenesis, the pentose phosphate pathway, activation of amino acids, fatty acid synthesis, the last part of the urea cycle, and nucleotide synthesis, to name a few, all occur in the cytoplasm. Additionally, proteins within the cytosol play an important role in signal transduction pathways. These proteins, found in the cytosol, also act as intracellular receptors and form part of the ribosomes, enabling protein synthesis. This concludes the BiochemSerye episode of the lecture on Introduction to Biochemistry and the Biochemistry of the Cell. Feel free to watch the other BiochemSerye episodes of this lecture as linked below in the description.